Okay, so continuing on the topic of inbreeding that we talked about at the end of the last one, how inbreeding caused by low effective population sizes is going to reduce the genetic variance or the heterozygosity of offspring. So more offspring are going to have more of the same alleles, so they're going to be less genetically diverse. And we're going to talk about the founder effect and how this can result in the accumulation of deleterious alleles, which is responsible for the population decline and eventual extinction that we see when there are population bottlenecks in certain species. So, at a low effective population size, the force of genetic drift overwhelms the rate of mutation, leading to more alleles becoming fixed. Pair that with high levels of inbreeding in isolated populations, and total heterozygosity further decreases, and lost alleles are unable to be regained by gene flow because the populations are isolated. This leads to what we call an inbreeding depression, where deleterious alleles, so alleles that reduce the fitness of an organism, so are typically negatively selected, these deleterious alleles increase in frequency as the force of selection is weaker than the force of genetic drift at low effective population sizes. These deleterious alleles tend to be recessive, and as genetic variance decreases and more offspring become homozygous, the more individuals will express the deleterious phenotype. This is called the founder effect, where rarer alleles are present at high frequencies in small isolated communities. So, for example, the pentadactyl limb in the Amish community that we're going to talk about now. But basically, you can imagine a small number of colonists moving from a large population. So imagine the largest population of Britain. You can imagine 30 British people. I imagine it was more, I'm just using this as an example. 30 British people emigrate from Britain to America and form an isolated colony where they only interbreed with one another, so they don't breed with anyone from outside the colony. So it's an isolated population. In the Amish community, one of these original founders who were part of the very first and very small effective population size had they were likely a carrier for a deleterious allele responsible for pentadactyly, which is where you have an extra digit on the hands or feet, so they have 6 fingers basically, or 5 fingers and a thumb if you want to be technical. So you can see here from the P generation, this would be the original colonists. You can see the carrier on the top here with the dominant allele for 5 fingers, and then the recessive allele for pentadactyly, which results in 6 fingers. So at first, it's not really a problem. The heterozygote carrier is going to breed with a normal homozygote, and 50% of their population of their offspring are going to be normal homozygotes, but 50% of their offspring are then going to be carriers. Now this is where the inbreeding thing comes in. So for the parental generation, it doesn't really matter. They're probably not very inbred whatsoever. You've got a carrier of the genetic disease, and you have a healthy individual. Now you can imagine 2 of their children, if they were to mate together, if they were to mate and they were to both be these heterozygote carriers, well, that's going to result in the next generation having only one of the healthy, normal, double homozygous genotype, and then you're going to get another two in that generation of carriers, which is increasing the allele frequency. So you can see in the parents, the allele frequency is 1 in 4 for the pentadactyl allele, the deleterious allele. In their offspring, this increases slightly to, sorry, it's 1 in 4 in their offspring again, the allele frequency, but then in the F1, the allele frequency increases to 50%, so 50% of the alleles in the F1 generation are this deleterious mutant allele. And then in the F2, if you can imagine these offspring breeding with each other again, so let's say this one and this one mate together, that's increasing the frequency of the deleterious allele again, and now the deleterious pentadactyl allele is greater than the normal allele for 5 fingers in a ratio of 3 to 1. So you can see as inbreeding goes from a normal parental cross to 2 of those siblings from the parents mating, and then 2 of those inbred siblings having children, and then their children interbreeding again. You can see the quantity of this deleterious allele responsible for the pentadactyly 6-finger mutation. That allele is increasing as the population becomes more inbred, so more individuals in that population are going to express the mutant phenotype of having 6 fingers. We can apply population genetics to help with species conservation. The method allows us to measure genetic variation in inbreeding, and such data can be used to guide breeding programs. Methods include heterozygosity, which can be measured from a single individual from the number of diploid loci that have different alleles, or het sites per kb, by measuring nucleotide diversity, which determines the population's average heterozygosity from the number of heterozygous positions from 2 randomly selected chromosomes. Through measuring inbreeding, which can be used to calculate the population of genetics of a pedigree, with chromosome regions being identical from descent. Genomes can be sequenced to find runs of heterozygosity, so runs on those chromosomes that are heterozygous, so different. These runs of heterozygosity can be analysed for long runs of homozygosity, indicating recent strong inbreeding, example brother-sister mates to give 2 versions of the exact same gene that came from their parents, or from distant inbreeding with short runs of homozygosity. So I just put kind of an image on the right here. Imagine the X chromosome, this kind of X, on the X chromosome you can see this blue region. Let's say that's the gene that we're looking at, always inherited on that chromosome that we've marked with the red star. As these 2 parents mate, both their children have ended up with this allele on each of one of their X chromosomes. And then imagine those children mate, and they have a daughter, and they've both inherited the X chromosomes from both of these parents again. they're more likely to have the